master of ceremonies, and Jerry Mead from the older folks who realizes the prime guru in the history of respiration that knows respiratory mechanics. So at the end of my talk, he came up and said, "That was the best last talk I've ever heard." I had mixed emotions here. I know exactly what he meant. So this actually may be my last talk, since I'm contemplating or actually have stepped into retirement. And I use this slide here. This is the male half of my family. These are my two sons-in-law and my grandson, because this is symbolic of this transition in my life. The rest of the family are much more attractive. It's my granddaughter and my wife. So it's a little personal background. And this is where I work. Give a better picture later. Now I'm going to start by looking at the ventral surface areas that Gordon already replied to. And I've been told that if I use this pointer, it'll be on the video. So I'm going to do my best to do that. We got that right? Sure. Okay. So these are the three surface areas. This is the surface of the ventrolateral medulla. This is a rostral, intermediate, and lateral area. And these are the people whose names were identified with those areas. And they're very interesting people. I have met some of them. I know that Jerry Mitchell worked in California, and after he did this description and established his name, he spent much of his career fishing after that. I know that Marianne Schlepke worked in Germany, and one time I went to a meeting in Germany where she presented her thoughts on chemoreception. She worshipped Hans Leshker, and I mean worshipped in the Germanic word, the Germanic sense of the word. We had a ceremony in a dark room with candles going where she talked about her relationship with Leshker. So there's a personal characteristic among these old giants that may be lost, except Jack does a pretty good job of that personal characteristic. So this is our first step towards trying to answer the question whether there are other sites involved in chemoreception. And this is Lee Folks, who works at Allegheny College now. And the idea was to make tiny injections of acetylcholine to block carbonic anhydrase inhibition and produce a focal region of acidosis. The idea being that if we produce focal acidosis in these different parts of the brain and stimulated breathing, they had to have a chemoreceptive element. And this looks at the pH as a function of distance from the injection site. And this is the measuring the pH in the extracellular fluid. And it's a fairly big change, but then it drops off very quickly. And this is just a series of cross sections through the medulla showing that there are many sites that seem to respond to this. So this was a deeply anesthetized animal, and we would like to avoid anesthesia for the following reason. This is an awake goat, and this is the level of ventilation when PCO2 in the arterial blood is increased from 40 to 60. The ventilation goes up fourfold from 5 to 20 in the anesthetized goat. And then if you anesthetize the goat, it's down here. It's a severely depressed CO2. So much of the work that we've done since then has avoided anesthesia. And the model that we've done then for many years is to produce a focal acidosis, much like Lee Folks did with the acetylcholine injections, but now using a microdialysis probe. Here's a cartoon of it, and the tip of this probe has a semi-permeable membrane. So the inflow has high CO2, CO2 flows out, and you have a little focal region of acidosis. The acidosis with this system is about equivalent to a 7 millimeter of mercury increase in PCO2. So it's very mild. It's much more mild than we did with the acetylcholine. And we are able to study the animal and measure EEG activity to establish their sleep state. We're able to study them awake and in non-REM and REM sleep and characterizing them based on the EEG criteria. This is the pH change we see here. This is the stimulation of ventilation we see during the dialysis period. And AOLE was the person mostly responsible for doing these experiments and should be the one taking over my main grant as I begin my term. So we use that same approach in multiple areas. So this is really repeating the Coates experiment, but now in awake animals using the focal dialysis with the pipette. And we find that there's widespread sites where there's response. This is a sagittal section then of the hindbrain here, and then the forebrain. This is a cross section at one of these levels. And each area in red here is an area where there was a response to focal acidosis in unanesthetizing. So this is a cross section. This is the facial nucleus, the raphe region. This is the RTN that Jeff alluded to. And these are people that have been responsible outside of my lab for some of these experiments. So we think there are regions in the cerebellum, the caudal NTS, the locus ceruleus, as someone brought up a few minutes ago, along the ventral surface, the preventitive complex itself, and this RTN region. We think all of these sites can potentially contribute to chemoreception. In addition, there's this area in the lateral hypothalamus which has a population of peptide-containing neurons. The peptide is orexin. Orexin is really a peptide that has many actions. It has a circadian rhythm. It's higher during wakefulness. It's sort of what you call the wakefulness hormone, and it stimulates many activities that we can think of. But it also stimulates breathing, and it's involved in chemoreception. It's also involved in regulation of blood pressure linked to chemoreception, and I'll come back to the possible translational utility of that observation in a few minutes. This just reiterates our new view on the right versus the old view on the left. And now I'm going to go into newer experiments that use molecular and genetic approaches to understand the role of these different sites and possibly the role of molecules within those sites in detecting CO2 pH and stimulating breathing. This is looking at molecular level. So we'll start with glial cells first. This is a paper that Alex Gorman published a few years ago. And the nice thing about these pictures up here is that the green cells are glial cells. And notice the location of these glial cells opposing this blood vessel, and this is the ventral surface of the medulla. So the Gorman group has been able to bring into the forefront the possible role of glial cells in chemoreception. So this is not only a different region of the brain, or possibly different regions of the brain, but it's a different cell type. It's not a neuron that's participating. So here's a model, and this is a picture of Mike Spire, who started this on the bottom, and Alex Gorman's picture. I've got to be careful what I say. I didn't have a picture of him available. So this shows a model of how RTN neurons and raphe neurons, which we'll come to in a minute, may be responding to substances released from glial cells in response to CO2 stimulation. And so here's a change here that's affecting an acid-sensitive ion channel directly in the RTN neuron. And it's also releasing ATP here that's going to bind to a P2Y receptor, and that's an ATP glial-specific mechanism. The same set of mechanisms may be participating here with a slightly different receptor subtype in the raphe. So the glial cells may be participating. We've done experiments in our lab where we poison glial cells and we can alter the CO2 response in the whole animal, which is supporting these ideas from England. So the neatest approach to this in my lifetime as a scientist, actually, is Susan DeMicki, shown up here, is one of the investigators and is now the leader of a program project grant that I've been working in for almost 20 years now on the sudden death syndrome. So this program project grant includes Hannah Kinney, who studies the tissues from babies who have died from SIDS, Susan DeMickey right now, who's a geneticist at Harvard, and a group of people at Dartmouth who look at the physiology of the animals. So we're working on animal models as well as human tissue. And the human tissue findings guide our animal model experiments. So I've had many, actually next Thursday we're having a meeting in Boston about this issue, with this issue. So Susan is an MD PhD by training. She has a lovely family. She works at Harvard Genetics, and she's a terrific collaborator as a person and as a scientist. And I explained to Susan that to me, the ideal experiment would be to somehow create an animal where we express the receptor in the cells of interest, the chemoreceptor, possible chemoreceptor cells of interest, which we could then reversibly silence by making a systemic injection. This was like a pie in the sky meeting we had a few years ago. So she did not create this DREAD system, but she used the DREAD system that was created in other labs and used it in mice to express in SERT-expressing neurons. SERT is the serotonin transporter. So the serotonin transporter is almost uniquely expressed in the serotonin neurons. And our goal was to express DREAD, is the HM4D receptor, in serotonin neurons, so we could inject the CNO systemically and turn off these serotonin neurons. That was the idea. So this shows in one model, these are GFP, which is a stain for the cells that are expressing GFP, and they're driven by a serotonin system. And here's the expression of the HM4D. The issue with the anatomy in many of these DREAD experiments is the transgenic origin of the DREAD. We have to express, they have to be done in parallel. So you can't express the DREAD receptor and the molecular marker, the cell marker in the same animal. But you can do them in parallel and come up with a pretty good idea of where your DREAD is being expressed. So we know anatomically where we're expressing the receptor. So this is Russell Ray, who is now an assistant professor at Baylor. Russell Ray, who's now an assistant professor at Baylor, and he did these experiments. He was working in Susan DeMickey's lab and learning the genetic techniques, and he would drive the mice up to Hanover in a special van because we wanted to avoid the incredibly expensive way of traveling with mice officially. So he would put them in his car and drive them up, and then he would do the experiments. So there's a lot of young people there. The thing that's impressive about Russell is he would bring these mice to Hanover, and he would do the physiology experiments with them. He would read into every experiment. He learned every detail of the physiology experiments, and he's taking all those details with him until he's in the position at Baylor. So here's the protocol. The animal is in a chamber. They're unanesthetized. Room air, 5% CO2 room air. You give the CNO systemically, and then you measure the CO2 response again. And here you have your animal where we silence the serotonin neurons all throughout the animal, and we see a reduction in the ventilation response to increased CO2. And then this is the sibling control. There were a lot of interesting experiments with the DREAD system along the way, one of which is they expressed the DREAD through all neurons in the brain and injected the CNO and depolarized all the neurons. It wasn't a cell-specific expression. And guess what happened? The animal died right in front of us. The other interesting experiment was when we did this experiment with Russell. We didn't know that there'd be change in temperature affected with this. We're measuring body temperature by telemetry in these little animals. And we keep them, when we do the experiment and the whole body plethysmograph, we keep the temperature of the animals well within the thermal glucose zone, the ambient temperature above the thermal glucose zone, within the thermal glucose zone. And the thermal glucose zone for these pups is somewhere around 27 to 28 to 30 degrees centigrade range, depending on their postnatal age. So when you take them out of the plethysmograph and put them in the room temperature is about 24 degrees centigrade. So this is one of these experiments that you walk in and you say, what's going on? And I've never forgotten this. We took this pup out, put him into room temperature, and his body temperature plummeted. It was like he was a Poico effect. We had made him a cold-blooded animal by expressing dread in serotonin neurons. So we can move on and look at different sources of serotonin neurons. And what Susan Danicki has done with her lab is to use what they call intersectional genetics, where they can do rhombomer-specific combinations of serotonin neuron silencing. So you have two drivers, one that's expressed by the rhombomer that you're trying to mimic, and the other which is expressing the serotonin activator that you're interested in. The EGR2 domain, the CROX20 domain, which is really around raphe, magnus, and RTN, was the source of this large effect here on the CO2 response. So it's not all serotonin neurons, it's just one set of neurons that are in the region of the rostral gullet. So Susan and other people in her lab have been looking at other subsets of these neurons, and the 5-HT neuron function depends really on the location and origin of the source of those neurons in early embryological life. So here we see the EGR2 PET1. PET1 is the driver for the expression of serotonin neurons. These are the ones that have connections to other chemoreceptor sites, other putative chemoreceptor sites, and these are the ones thought to be involved in chemoreception. In the more caudal region, the R6-7 organ, these actually project mostly to motor nuclei, respiratory motor nuclei. And this group would be of particular interest, I think, in the Gainesville group, because they're the subset of serotonin neurons that might be affecting the processes that we heard about today. So that's the serotonin neuron group. Now, orexin, I put up here as a reminder of these dollar signs. It turns out that orexin and things that inhibit orexin receptors, like omorexin, which is a drug that we use to inhibit orexin receptors, we thought that this was something that was new and exciting to us until we read a New Yorker article maybe a year ago, describing how much money Merck has spent looking at omorexin or orexin receptor antagonists. We get our orexin receptor antagonist by a relatively small potatoes until a few months ago company in Switzerland who was very nice to send this to us. And it turns out it's probably been over $10 to $20 million spent by drug companies trying to develop orexin receptors that don't interfere with sleep but do have the physiological effects that I'm going to tell you about. So what Mirella Diaz found, she's from Brazil. This is the guy from the shadows there. You know Mirella? Yeah. Okay, so these are where the orexin neurons are, and they project down here to the raphe, the medulla, and the RTN. And we can get focal inhibition of the orexin 1 receptor in these regions and reduce the CO2 response using this orexin 1 receptor antagonist. So by blocking orexin inputs, we can alter the CO2 response. One idea is that we have, and we haven't really studied it directly, is the CO2 response in all of us is depressed during sleep. And that may be explicable entirely by this orexin link, because orexin releases is diminished in sleep as well.
Sure, and serotonin too.
It may be part of the serotonin. If we give it systemically, we inhibit both receptors, we also see a reduction in the CO2 response. Okay, so we think orexin is playing a role. I'll come back to orexin briefly. Well, why don't I do the orexin thing now? I think one of the interesting things is in the what's called the SHR rat, the spontaneously hypertensive rat, and that's supposedly a model of neurogenic hypertension, the increase in blood pressure that occurs from excessive stimulation of the sympathetic system by if you block the orexin receptors with this Swiss antagonist and SHRS, you see a return of blood pressure almost at point. If you take the juvenile SHRS, this is unpublished as of yet, and you block the orexin receptors with almorexin, blood pressure comes back to normal. So the increase in blood pressure in this model of sympathetic hyperexcitability is probably related to orexin. And that's why these companies are spending millions of dollars finding orexin antagonists. Because if you can treat high blood pressure with a drug like almorexin without interfering with sleep cycling, that would be a money waste. Okay, so Jack Feldman has described the melatostatin receptor system to you, and a whole bunch of people here collaborated to use Jack's methodology, actually. And this time, the melatostatin receptor here was expressed in RTN neurons here just below the basal nucleus. It's a series of cross sections from the rostral, and here's the FOX2D marker for RTN neurons, and here's the EGFP with the melatostatin, and here's the WA. So if you block the function of RTN neurons by injecting the melatostatin in this region or into the cerebrospinal fluid, you also can affect the CO2 response. And this is Jack in his young day, maybe, and this is his friend Julian Payton. And this is the diaphragm amplitude times rate. This is a measure of ventilation. And this is what happens after blocking the melatostatin receptors in the RTN neuron. So it's pretty clear the RTN is a pretty important chemoreceptor. So the subsequent debate, which has a lot of personal or personality overtones, is the RTN the most important chemoreceptor sites? Is it by itself the most important? Or do all these other sites that can talk to the RTN, can they independently change the CO2 response or modulate it or be responsible for it? So there's a person named Patrice DNA. How many people have you guys met Patrice or read his papers? Well, he's a champion of the RTN being the most important chemoreceptor site. Other people are less enthused about how whether it's the sole and the only important site. So that's part of the controversy. I'll show you a little interesting data about that controversy. So this is Garitas, a French investigator. This is young animals, P2, P8, P22 in adults. This is their response to 8% CO2. And these are conditional knockouts for something called Ondine's curse. Is there anyone that has not heard of Ondine's curse? Okay, you have not heard? Okay, so Ondine's curse, it's basically called, it's a name that John Severinghaus gave to us, an interesting set of patients who in the first years of life hypoventilate, retain CO2, especially in sleep, and they will eventually die from inadequate ventilation. And these patients have to be treated in pretty invasive ways. They have to be ventilated with ventilators, especially at night, and they can actually live to be adults and have children, procreate, if you will, as long as they have a ventilator to treat them at night. And I've been to the meetings of the CCHS club, if you want to call it a club, where there's probably 20, 30 patients there and a lot of scientists that are interested. And they're a very interesting group of people because they have to be on a ventilator every night when they go to sleep. They cannot forget to breathe. Well, Ondine's curse was a curse in mythology where Hans, the lover of Ondine, the sort of semi-god or semi-human man, was cursed to have to remember to breathe when he went to sleep because he had been unfaithful to Ondine. That's the curse. So anyway, so the EGR2 targeting of the FOX2B mutation does go away with the response to CO2 here, but then it returns when they become adults. So these data suggest in a conditional knockout with FOX2B disruption with EGR2 targeting, that the RTM is really the big player in the CO2 response. And I would say the data are fairly convincing that in early life of rodents anyway, it is the big player. So a paper came out from Patrice's lab at Virginia, DNA's lab, and Kumar's, she's the first author. And this is called Regulation of Breathing by CO2 requires the proton-activated receptor GPR4 in RTM neurons. Now we're going into the molecular level. So they were able to alter with knockout mice expression of GPR4 and or TAS2. TAS2 is a potassium channel modulating protein that it's also been thought to be participating in chemoreception, but only at higher levels of CO2. So this cartoon is not from their paper. This is a cartoon that I drew on a little scrap paper and sent to the people at the New England Journal, and they created something like this. They had asked me to write a commentary on this science paper by Kumar. So the title that I came up with is On Being Undone. And these studies suggest that the GPR4 knockout has a reduced CO2 response here compared to normal. And then if you knock them both out, the TAS2 channel and the GPR4, you get a response that they say is non-existent. So this is the cross-section that we've looked at. This is the neuron showing you where these receptors would be, and here's a cartoon with the receptors. And I'd like to add one more other piece of information that wasn't part of the paper, and that is that if you look at the sequence of amino acids for the GPR4 receptor, there are histidines in the active site. Histidines, and I'll tell you that story briefly in a few minutes, have been proposed by comparative physiologists to be the proton sensing site that could be explaining chemoreception in animals who live at vastly different temperatures. So pH, if you don't know it, changes with temperature. So if you go out in Hanover anyway, you find a frog when it's 25 degrees out there, that frog's temperature is going to be very, very different. As the temperature goes up, the pH goes down, and this is because of the effects of temperature on the dissociation of water, H2O to H plus and OH. And if you have other molecules that are pH sensitive, they will also have this kind of temperature pH relationship. So in Hanover, on a regular cool day, the frogs that are hopping around happily really have a body temperature that's quite alkaline. So alkaline that in human eye it would be dysfunctional. So we can explain that. I can explain that in a minute. So the idea that's of interest to me, since I've always been interested in this comparative approach, is this GPR4 receptor does have histidines. And here's the idea that you block that receptor, you get a reduction in the CO2 response. You block both receptors, and you get very little CO2 response. So Dr. DNA Patrice is taking this as further evidence that the RTM, which is where these receptors are in the RTM neurons, is really the key site. So, here's Patrice and a happy day. And this is a paper where there's a review he has written. And here's a cross-section, much like the ones that Jack has shown and I've shown. And here's the RTM. Okay? And here's the efferent projections to the RTM. And here's the GPR4 receptor and the TAS2 receptor here. And Patrice's idea is these other chemoreceptor sites really project and converge on the RTM. So when we change serotonin function in rat-based serotonin neurons that affect the CO2 response, that's because those serotonin neurons aren't stimulating the RTM as much as they do you know, it's really hard to, it's really hard to come to a conclusion about the answer to the question, so what? What does it really matter? Well, I think it may matter in certain disease situations where the CO2 response efficacy is quite important. So for CCHS children, it's very important to be able to figure out how you might avoid their life of being on a ventilator. In the review I wrote here, I had to come to the conclusion that the editors agreed that this did not, they changed my sentence, that this was not going to be likely easily achievable in the near future. That's the same molecular way. But when I listened to all the talks here in the past day, it seems like you could, and especially what David was trying to do, you could possibly entertain the idea of a viral construct that would affect RTN neurons and chemosensitive function. But again, this is like an orphan disease. I don't know how frequent it is relative to Pompe's disease, because it's not very frequent. The difference between Pompe's disease and SIDS is every mother worries about SIDS, even though there's probably 2,000 SIDS deaths a year in the United States. But every mother who's read anything worries about it. So there's a lot of anxiety related to SIDS that could be alleviated if there were a therapeutic avenue that was inexpensive and suggestive. So here's Patrice's view. And here's his view of the system even simplified more. Here's the RTN with raphe, hypothalamic, orexin, NTS. I didn't talk about the NTS, but J.D. studies with NTS neurons were really the first clue that there were other cells in the nervous system that were very sensitive to pH. That sort of triggered my interest in multiple sites some years ago. So here's Patrice's view. And note, in this interpretation, all roads lead to Rome, aka the Roman trapezoid. This is a way to remember Patrice's view here. Okay, this is an alternative view. This is again from a paper from Susan Demicki's lab, and this is Rachel Roster's excellent PhD student. And this shows, again, a cartoon with the RTN down here. And guess what's dominant in this cartoon? It's the raphe neurons, because that's what she was studying. So here she's got raphe neurons projecting everywhere and affecting function at these different sites. So this is a cartoon view of how we can look, and the difference in the next two slides, which are mine, and the previous slides, is that they're differentiated according to state. And this is hypothetical. So in wakefulness, the orexin system is functioning, the locus ceruleus is functioning, the NTS, these are the chemoreceptors, as this should be read, and the central pattern generator and the pre-Betzinger complex are here. And so the RTN is part of the system, but it's not the primary integrator. And then when you go from wakefulness to sleep, well, this isn't playing anymore. The raphe isn't really playing anymore. And we have a system that has far fewer components that are accessible to CO2 responses. So this is not an accurate depiction, but the idea that there's a different circuit, I'll use the term circuit, a different circuit in control of breathing and wakefulness and sleep is not a new idea. So if we take an anesthetized patient or animal and hyperventilate that patient or animal, so we can have a patient and bag that patient, but they're anesthetized or they've had head trauma, and you bring the CO2 below normal, they will stop breathing. They will have apnea. But if I start saying, okay, I want all of you to hyperventilate with me together, we'll do this, we can all lower our PCO2. None of us will be doing that. So the system is very different in wakefulness than it is in sleep, at least in terms of what the CO2 is doing. And this can explain some of the data in some of the mouse models that I'll show you in a second. So this is a recent paper from the Brunet, this is the French group. And this is showing that the retrotrapezoid neurons express ATO1 and FOX2B. And again, this is the response to CO2 in a P0 animal is absent. But when you get later P60, here's a response that's present. This is roughly like the last set of slides, 3 or 4 slides I showed you. So you can disrupt the RTN here, not by disrupting the channels that are thought to be chemosensitive, by disrupting ATO1 as well as FOX2B, which are really factors that are determining these neurons in your face. So here's the quote from that paper. So these guys are sort of straddling the fence, I think. A consensus about the sites and circuits that underlie the chemosensory control of breathing has yet to emerge. Our results establish that the FOX2B ATO1 on RTN neurons, that is the RTN neurons that have expressed RTN in embryo, lie at the core of the chemorequest circuit. That's it. You know, if you know the field, that's really a very carefully worded sentence. But they're sort of saying, okay, RTN guys are important, but there may be a little bit more to it, especially when you get older. So, I just want to reiterate something I've skipped over, and that is if you look at which cells are sensitive to, in slices or in vitro, to pH, RTN neurons, serotonin neurons are, JD, NTS neurons respond, orexin, Vertikov, and at Oxford has shown that orexin neurons are very sensitive to CO2. And then norepinephrine neurons have been described as sensitive, and Patrice has looked at RTN neurons, and we have these glial cells. So there are many elements that, when taken out of the system, can respond to pH changes. The issue to me is, when you put them all back in the animal, how do they interact, and under what circumstances do each of them play a role in breathing? So I won't go through the Alvestat hypothesis, which I mentioned briefly. This is the histidine dissociation. This gets back at what the role of those histidine molecules in the GPR4 receptor would be. But the idea is that when pH changes, the fractional dissociation of histidine changes in parallel. So the protein conformation state is unaffected when pH changes due to a temperature change. So when temperature changes, pH changes, and dissociation of histidine changes in parallel, so the protein is happy. But if you're at a constant temperature like we are now, and we make you have an acidosis, then it's a different ballgame. You're going to stimulate the response of the receptor cell via histidine mechanism. Okay, that's just one. So this is where I work. These are the people that I work with. The Geisel School of Medicine at Dartmouth is named after Theodore Geisel, who wrote, what did he write? Dr. Seuss, The Cat in the Hat. He was a Dartmouth grad, class of 25, and he supposedly gave the medical students, medical school, lots of students, $200 million. So as soon as that money came in paper, I don't know if I'm supposed to tell this story, having said it, but so they renamed the school in his honor. His wife hasn't died, and he has children that don't agree with this, and we don't have the money. So the school's named after him, but I don't know what's going to happen.
Did they consider calling it the Dr. Seuss?
Yeah. Let me ask you, I know you love it, Dave, so don't get ahead. I think you're the first person to suggest Okay, and these are actually the pictures. It's nice to show pictures of the folks. That's Hannah Kinney. I know you guys all know George and David, and I know a little bit about Jack from listening to us. I don't know if anyone I've ever met who's dedicated trying to get people to work together, trying to get tissues. All of the tissues she studies in terms of human tissue for sudden death syndrome come from California. You know why they come from California? Because Sid's parents in California got the legislature to pass a law saying that infants who die without an explanation, their tissues can be used for research without parental approval. Okay, there's Susan. She's also a very impressive person. Ayla, who you see, Russell, Stella Lee is a current student. She trained with Bill Nelson and then has come to Garnet. Kevin was a postdoc. He's gone to Missouri. Andrea was a Harvard Francis Fellow, and she is now teaching in a small college in Vermont because she wants to stay with her husband who makes beer in Vermont and her child. She's a very talented person. Hard to move a brewery, huh? Carlene did experiments that I didn't tell you about. That's Rachel and Mrs. Ryan, who's home to Johnson, who's an MD PhD student, looking at dread experiments that he's done with Andrea on our resuscitation in infants. That's the SIDS laws. I says, yeah, thanks very much. Jack had it first, and then.
Right. Gene, one of the problems I've always had with this notion about neurons being CO2 sensitive is the magnitude of the stimulus that's the look at responsiveness. So a 1 millimeter change in PCO2 produces a 40% change in methylation. So it's relatively small change in
The whole animal, right?
The whole animal.
And that's exactly one of, that along with J. Dean's data was one of the things that thought that made me think there has to be a network.
But the data you showed is like going from 10% inspired CO2 to 5%, or George is using 5% to 0. I mean, those are, or pH 7.5 to 7.2, which is a threefold change in hydrogen ion. And are you able to scale the responses down? So if you have a 2% change, that you get a change in neuronal activity that can account for the huge changes in ventilation?
Right, so the way to do the experiment runs into the issue we talked about, I think, earlier this morning about the noise in the system.
Yeah.
So if you want to give 2% CO2 to animals, you will be able to detect a ventilatory change to that, but it's much harder to.
No, I'm not saying 2% CO2, I'm talking about 2% change in CO2. You know, from 40 to 41 millimeters of mercury, you get a 40% increase in ventilation.
Right, so the original experiments by Pappenheimer are the best experiments. Pappenheimer did ventricular neural diffusion and away it goes. And he was able to change the pH in the brain and change LDL, produce a ventilatory response. And that was a very sensitive system. I had those data.
But he was talking about a whole animal data,
Whole
and
animal.
I'm talking about
I know.
the assertion that single individual neurons in a given part of the brain are CO2 sensitive,
Right.
but that's based on huge changes.
But to get an individual cell in culture to respond at this sensitivity level, no matter what Patrice says, has to be.
So I'm very skeptical, not cynical about a lot of that data. Your response is, well, but if you get enough of these together, that adds up to
Well, Patrice's answer, I think, would be that RTN neurons are that sensitive. But I don't see.
I've never seen that.
I've never seen that.
No.
So Jack's basically saying that the cells that are studied in vitro, cells are in some isolated way, you require a bigger pH change to get the change their firing rate than you do in the whole animal to get the ventilation to increase. And that is it, right?
Yeah.
To be fair, you know, in a rodent, they don't respond as much as, let's say, a goat. I've studied goats over the years and people. They're very responsive to what you're talking about. Unbelievably responsive. Rodents are less, but your point is still a really good one. It's hard to come up with even that sensitivity, looking at these neurons and the changes you see. That's a game, so I think that's a good one. Brendan, you had a question?
Yeah, this relates directly to what you just said. If orexin is so important in chemosensation, orexin is directly related to circadian rhythms, and rodents are nocturnal.
Yeah, so you have to study them at night or versus, you know.
Right.
Their awake period is at night.
Right.
So we have a lot of 24-hour experiments.
So,
So we know that. And when we want to study wakefulness, we have to do it in what we really do is try and make a light dark cycle that allows us to study them in the dark during our day. So we put them in the animal chamber, change the lighting, bring them upstairs, we cover the cage, and so we can study them in their dark periods during our day.
Has anyone looked at other things relating directly to sleep circadian rhythms, like histamines or melatonin levels?
We've looked at MCH, that's melanin concentrating hormone, which also participates in chemoreception. That data was recently published or it's in the press, and I didn't include it here. But that's another lateral hypothalamic peptide that participates in modulating chemoreceptor response. The other two that you mentioned, Gordon mentioned that serotonin, all the monoamines have more or less a cyclical change in their release. So I don't know if people have examined that as directly as we have done in
Question. I've always liked the dread approach to those experiments. But one thing troubles me a little bit in that, and that is that CERT is also in glial cells. It's in astrocytes. So what would happen in this context? Was there a way to restrict it to neuronal drug receptor expression, or is it going to have the glia too? Because then you cross over into the green and spire kind of argument.
Yeah, no, so there is a problem even with the dread system in the sense that whatever you use to drive your dread receptor expression has to be cell-specific.
Right.
So if we look at PEP1 versus CERT as the serotonin driver for H114 expression, the PEP1 and CERT distribution is not the same. And if you do, I haven't done this, but Ben Okadi was doing this in Susan's lab. They do single cell RNA-seq looking at serotonin neurons. And they find quite, it's like JAX data from the pre-Betzinger conference. They find a large variety of subtypes of neurons, which are all serotonin neurons, but they're expressing very different amounts of different genes and peptides. So,
But the glia would have sort of an unpredictable response. Right? You could close a few
that's the dirty part of even the dread experiment.
Yes.
It's the cleanliness of your driver.
Another question, then go back there. You know, when you do, let's say, a dread experiment or an elatostatin experiment or whatever, you shut down neurons in a putative central chemoreceptor group, and you see the CO2 chemo reflex that's reduced. How do you separate out that the chemo sensor has been removed versus it's something that modulates any step along the chemo reflex?
So that, you sound like Patrice here. Patrice's answer to all questions regarding non-RTN neurons would be what you're asking, that the effect on the whole animal systemic CO2 response is to alter the modulatory input to RTN.
Well, I don't mean to go to RTN, but I mean, there's a lot more to a chemo reflex, right? Yeah,
No, no.
including the motor neurons.
Yes, I was making a narrow answer to your question, but you can broaden my answer so it would cover all possible interactions.
Yeah. Well, and then there's the Richardson experiment that Matt Hodges did, where in their LMX1B knockout mice, they're hypo-responding in the same sense. But when they put in serotonin tonically, replaced the serotonin with an LZ pump, the CO2 reflex was restored. That's not a chemoreceptor thing. That has to be modulation, right?
It is modulation.
Yeah. It's just hard to separate.
Right.
Other questions? Yeah, Dave.
So, I'm very fond of this. What would be the, what type of data would we need to accept one hypothesis versus the other about the role of the RTN as being more integrator, like Dr. Guinea says, or what would your model, like what's the data that would be most useful?
I don't know, actually. I mean, some of the things that Patrice and the French guys say, I mean, that quote I gave you from the Reed's paper from 2015, that's a reasonable quote. I think in young animals, the RTN does seem to be crucial. As you get older, it means that these other sites may be kicking in a bit more.
Or are they kicking in yet more because the RTN's been removed? Kind of like the Kurt Forster lab, with the innervated carotid body and the aortic body.
Yeah, it could be something like that too.
Yeah.
So you could have a modulation of a secondary effect by removing the primary source.
Also want to ask, when you did the red cert experiment, did you look at what happened to sleep? Is that possible?
We did not look, it is possible, but we did not look at sleep in those experiments. So those were done at Dartmouth, but they were done by people coming from Harvard to share experimental expertise in learning with Dartmouth people. So we didn't really do EEGs in sleep. Great point.
I'm curious if you can see more
Yeah.
happening there with structure.
When you do the orexin antagonist experiments, we do measure sleep, and we do see more happiness. And that's the scary thing to the drug companies that want to promote orexin antagonists as an antihypertensive. If you block the blood pressure, bring it back to normal, but the patient has more sleep apnea, I mean, who's going to win, right?
Thank you very much. For those that would like to talk to Gina and Jack a little bit more, we're going to head to Tall Falls. It's a little bit of a shift in our, where we were planning to go, simply because the weather forecast didn't look too good. So come on down and join. There'll be some beer and some pizza. You see a pop out on top of that, it's different here. I'm like a 10.